In this video, we will discuss about these KRAS mutations and the T cell priming from this pancreatic cancer cells perspective. So in the pancreatic cancer cells, around 92% of the cases, people have found out that there is a mutation in the KRAS, and KRAS belongs to a family of this RAS, where it includes not just KRAS, but it has this 3 different GTPase or 3 different RAS GTPase. It is KRAS, it is KRAS, HRAS and NRAS. Out of this, KRAS has found to be mutated in most of the cases in the pancreatic cancer cells. And in normal cases, what happens, this KRAS regulate the GTP bound inactive state to GTP bound active state. However, once it is mutated, in most of the cases the GTP bound active state remains stuck up, and this GTP bound active state, what it will does is it will trigger the downstream signaling towards proliferations, migrations, that is by triggering these survival pathways, thereby preventing the cancer cells to trigger this programmed cell death. This is one of the reason how this pancreatic cancer cells continues to grow. And now we'll discuss how it is related to this T cell priming. So once this KRAS is mutated, then it will cause this GTP bound active state. It will cause this RAS to remain in this GTP bound active state, and this will result in the mutations within the cells, and that will trigger this cytosolic protein towards this MHC-1 complex or peptide MHC-1 associations. And this peptide MHC-1 associations, and this usually happens because the mutations in this KRAS will result in the formation of peptides that are foreign or that are not normal to these pancreatic cells. So that's why it's trying to express this cytosolic protein or the peptide of the cytosolic protein on the cell surface membrane. So the process occurs with this cytosolic protein will go via this endogenous peptide formations through this proteasome, will enter this endoplasmic reticulum, where it interacts, where it form a peptide MSC associations. In this peptide MSC associations, after that in the non-silated peptide MHC will transport it to the Golgi, since silation is not occurs in the endoplasmic reticulum. And in the Golgi, in the normal case, what will happen, that this in the MHC there is N86 region in the heavy chain. It is glycosylated, but it is not silated in the normal case. And in the normal case, when it is not silated, then it can this N86 helps in the interactions with this T cell receptor, and thereby enables this peptide informations transfer to the T cells, and the T cells priming will occur, and thereby the T cells will form antibodies against those peptides. However, in the normal case, there is no peptide needs to be transferred as the pancreatic cells are performing normally. But in the case of the KRAS mutations, what will happen, it will also affect this glycosylation. This glycosylation is a very sensitive process. Even small changes within the cells cause changes in the glycosylation. The changes happens at the N86. Instead of just only glycosylated, it will also get silated within this Golgi, and because of the silations at this N86, it will not be able to interact with this T cell receptors, and the interaction is, if it is not proper, then this priming of the T cells or the peptide information transfer to the T cells will be weakened. And these things are being tested in the in vitro, where what the researchers has done, they treat this tumor cells with this neuraminidase, so that the salic acids on the cell surface can be removed, and thereby it is found that it will improve the T cell priming when they co-culture them with this CD8 plus T cells. So this is how this KRAS mutations in the pancreatic cancer cells is related to this T cell priming, and how the mutations affect the T cells priming. So this is one of the reason how the immune cells, how this not immune cells, this tumor cells is able to evade the immune cell response, by that the T cells will not be able to generate antibodies against the tumors because of the weekend T cell priming or the weekend peptide information transfers from the pancreatic cancer cells to the T cells. And the paper that we have discussed previously about Sialylation, what it, in that case, how they are able to improve the T cells activations is that instead of this T cells direct interactions, they enhance the phagocytosis immune response against the tumor cells by preventing the Sialylation and the ITGA3 and ITGA1 interactions. And then phagocytosis then provide the peptide informations to the T cells, and that increase the T cells activations against the tumor cells. So there are different ways that researchers has found out to activate the immune response against the tumor cells. So I hope you understood this relationship between KRAS mutations and T cell priming. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description, and you can also check the previous videos where we have discussed about this Glycan and ITGA3 and ITGA1 interactions between the macrophages and the pancreatic cancer cells respectively, and by blocking those interactions, how the researchers has increased the phagocytosis effect against the tumor cells and also enhances the T cells mediated cytotoxicity against the tumor cells. So that's all for this video. Thank you for your time.